there was a paper published a while ago with a hit-and-run hypothesis in type 1 diabetes, and they hypothesized that it was waves of antigen presentation by endothelial cells. That caused
Yeah,
that.
and it could well be, you know, this fits in really with chronic autoimmunity, with, you know, diabetes, with other kind of systems. This is you know, and it's that link that I think is really quite interesting from my perspective with those other kind of disease situations. So the immune dysfunction that we see late on after radiation is related in part to a lot of T cell activation. And although you get T regulatory cells coming up as well, you get both, you know, in balance essentially, or in imbalance if you like. And I think it's the way these kind of interactions take place in an activated immune system which determine the kind of final outcome. But it's fascinating to think that maybe autoimmunity is playing a major role in some of these late effects of radiation.
I just was wondering, at one point, molecular signatures were discussed. Is there an understanding of gene signatures early in these cascades and later on, if they're the same or different, and what kinds of transcription factors might be involved or something?
And that's exactly where I hope that all of that program is going to go, exactly there, because I think that we know a lot about the immediate early gene expression, which within the first 24 hours, we're just learning from these gene signatures some of the later 24 to 48 hours. Now the question is how does that develop? And I think the whole kind of, this relates to chromatin structure and you know, all kinds of many other really interesting biology within that. But I think it's, yeah, and I'm sure it does change with time, even though some of the gene signatures recur. I think they're a little bit different with time, and it's going to be very interesting to look at that. But I don't know the answer, but I know the question. I think it's a very good one.
Yeah, it's very interesting to think about it from this concept of resolution of inflammation, what drives it and why it's not able to occur.
Exactly, and that's one of the things which I hope is one of the spin-off from some of the work that comes out of the late effects of radiation, is how do you control some of these more chronic inflammatory situations. You know, we do have some kind of ways to do that now in the clinic, but we don't have all that many, and our understanding, I think, is still pretty limited. Thank you.
Thank you, Bill. I am going to move on to the descriptive part of our program. I didn't see it. All right. Here we go. So thank you, Bill, again. That was a very comprehensive yet easy to digest coverage of the physics and biological background for radiation and radiation injury. I am going to open up our conversation about the different elements that are present in our portfolio in the Radiation Nuclear Countermeasures Program, or RNCP. We have, this is a slide you've already seen, presented by Dr. Bateman, and the four of us in the program divide and slice this very busy portfolio. My part of the portfolio consists of the CMCR's U19s that Bill so eloquently described, as well as a project with the RERF Research, Radiation Effects Research Foundation of Japan, looking at the effects, late effect of acute radiation in the A-bomb survivors, in conjunction with intramural researchers at the National Institute on Aging through an intra-agency agreement. And part of my portfolio is also a suite of 6 R01 grants on predictive biodosimetry. So to launch into the program, Centers for Medical Countermeasures Against Radiation, the program was established in 2005 and underwent one first renewal in 2010. The current incarnation of the program includes the University of California, Los Angeles, which is headed by Dr. McBride, as well as Duke University, headed by Dr. Nelson Chow, who was a recent member of this subcommittee, as well as University of Pittsburgh, University of Rochester Medical Center, Dartmouth, Columbia, and Albert Einstein, 7 in all. The mission of the CMCRs from the very beginning and currently is to support both basic and translational research to develop products for the assessment, diagnosis, mitigation, and hopefully treatment of the consequences brought about by radiation exposure after a radiological or nuclear accident. So clearly there has been quite a bit of basic research activity. Bill said that his group alone is over 100 folks, and I can tell you that the other 6 are as well quite numerous. Up until this point, in having completed the 8th year of existence, after a very short period of infrastructure setup, 400 peer-reviewed papers have been produced, over 400 peer-reviewed papers have been produced. And in terms of product development, candidate compounds in the tens of thousands have been screened, and about 125 of them have been tested for the mitigation of ARS for the hematopoietic, gastrointestinal, and lung syndromes. Also, patents have been filed. 20 are provisional, 8 issued, for a total of 28 patents. And finally, of the candidate compounds that have been screened and tested within the CMCRs, 6 candidate medical countermeasures are now in BARDA's portfolio, together with 4 biodosimetry platforms. What I will do now in the next few minutes is to highlight a couple of candidate compounds that are of particular interest, and Bill has already touched upon. Also, one platform, biodosimetry platform, and a very interesting late effects of acute radiation syndrome, also carried out in our program. So Bill, as you recall, discussed John Schutz, the Duke and now UCLA, discovery of pleiotropin, which is involved in angiogenesis, osteogenesis, and ES cell growth. And what a key experiment that Dr. Schutz carried out was exposing mice to 7 gray of irradiation, which in this particular experiment causes about 50% death in a period of 30 days. Treating these animals with pleiotropin either at 2 days after exposure or 4 days after exposure clearly brings quite the survival benefit. And in fact, treatment at 2 days brings about 40% increase in survival, while treatment at 4 days, still a 35% survival, something that is quite significant. The current status of this program, of this particular project, is moving, it's a move to the BARDA portfolio, and of course, publications including in Nature Medicine under consideration paper. Another interesting compound is SOM230, also known as Signifor, which is marketed by Novartis. And these are studies conducted in the Duke CMCR by Dr. Martin Howard Jensen. In these experiments, Dr. Howard Jensen treated the mice with radiation and then either give them the SOM230 at 24, 48, or 72 hours after total body irradiation. And again, this off-the-shelf drug that is used for Cushing's disease was able to protect the mice after from death, at least within the 30-day observational period. when administered 24 hours, the survival afforded drug is an 80%, 2 days after, 50%, and even 3 days after, we're still seeing a 25% increase in survival. These studies, this particular compound has been in advanced development stage at BARDA since 2011, and it's well underway toward FDA approval, and together with Novartis, Dr. Howard Jensen has completed a pre-IND meeting with the FDA within the past year. I would like to move now to the rabbit. You've heard again from Bill about this tool. It is the Rapid Automated Biodosimetry Tool. And this is a platform developed by Dr. David Brenner, the PI of the Columbia University CMCR. And what is very important and interesting about this is that it automates very well established what is considered gold standard, state-of-the-art biodosimetry tools such as gamma H2AX foci measurement or micronuclear or dicentric chromosomes, which are, as I mentioned, state-of-the-art, gold standard, but unfortunately they cannot give biodosimetry results within the time frame that would be necessary right after an accident. We're talking about few dozens at best in a week, whereas we would need in the number of thousands per day of results. And so this system proposes to be fully automated with ultra-high-speed robotic biodosimetry with a capacity of 30,000 samples per day. The investment of the ground crew in terms of obtaining samples from the people affected by potential radiation exposure is minimal. It's in fact just one finger stick of blood. And what's really important is that once that finger stick of blood is collected, it enters the automated system and there is no further human intervention necessary. This platform is also now within the BARDA portfolio for further development of the automation. So the main technical innovation where, of course, the use of small samples, but again, it's the complete full automation of biology with in situ imaging to produce a system that will give answers within 2-3 days after an accident instead of having to wait at least a week after the accident and the sheer number of samples that can analyze. I would like to move now to a very interesting set of studies that is coming out of the Duke University CMCR at the Wake Forest Non-Human Primate Center. This center houses some, right now, 34 and still increasing number of non-human primates that have been retired for radiation studies conducted at the University of Maryland, AFRI, and the University of Illinois in Chicago. Each bar represents one monkey. The limit of the bar to the left is age at irradiation. The one to the right is the current age or the age at death. And as you can see, different cohorts of NHPs have been exposed at different levels of radiation, but I would like to make it clear that these NHPs never received any treatment. So they were part of the control cohorts of the monkeys that were used at these centers. And so these monkeys are monitored daily for viability, for any appearance of disease or any physiological abnormalities, as well as weekly, monthly, semi-annually, and annually for all sorts of different physiological functions, cardiovascular, kidney, digestive, immunological, neurological, you name it. It's quite a varied screen. And now that this center has been in place for over 2 years, the data are starting to come in, and in fact, I understand that some of these are in ready, are in preparation for publication. And so Dr. Mark Klein, the PI of this particular project, observed clearly anticipated effects that have to do with increased frailty, impairment in the immunological system, cataracts, renal injury, malignant neoplasms, but also a number of unexpected observations that had to do with late onset of myocardial fibrosis, diabetes, pulmonary lesions, osteopenia, and brain lesions. And so what I would like to draw your attention to is that in conclusion of the studies conducted thus far, what appears to emerge is a complex pattern of disease occurring at doses lower than anticipated, and they most likely include persistent inflammation and peripheral insulin resistance, to go back to what Bill was describing earlier on. This is a very interesting study, and it provides a very nice segue into the next item, the next element of my portfolio, which are the studies of immune senescence and other late effects of acute ionizing radiation exposure in A-bomb survivors. This is a program that was established in 2005. We're now in the 5th year of a study, and it is headed by the Radiation Effects Research Foundation in Hiroshima, Japan. This organization, this is a nonprofit foundation that has been in place since 1948 out of a collaboration between the Japanese and U.S. government. And it is both at Hiroshima and Nagasaki. The purpose of this program, the overall RERF, has been to follow the late effects of acute radiation exposure in the people that survived the blast at Hiroshima in 1945. And to stay within the spirit of this binational collaboration, our program, which happens to be a contract, isn't just with RERF, but also with 4 additional Japanese institutions, Tokushima, Chiba, and Keiyo universities, as well as the Japanese National Institute of Health Sciences. There are also 5 groups that are involved in this project in the United States, with the universities of Georgia and Arizona, Duke University, Memorial Sloan Kettering Cancer Center, and intramural researchers at the National Institute on Aging. As I mentioned, this program has been, the general RERF program has been in place since 1948. And in fact, the in utero study was started right after even before the inception of the particular program, looking at the effects of radiation in individuals that were in utero at blast time. 1950 marked the beginning of the lifespan study that enrolled 120,000 people and is still ongoing with the survivors. But of interest to us is the adult health study, which at inception in 1958 enrolled, and through the 60s and 70s enrolled 23,000 people, and is also ongoing. This study is of particular interest because, contrary to the lifespan study or the in utero study, it concentrates on monitoring individuals with biannual visits, during which all sorts of different physiological parameters are being examined and blood is collected. And it is focused on assessing the long-term effect of acute radiation exposure on non-cancer related disease, both morbidity and mortality. The goal of our program with RERF is to define the long-term effect of acute ionizing radiation on the aging of the immune system, human immune system, which we know already that radiation exposure brings about effects and syndromes that are very much like aging of the immune system. So we wanted to know how much those effects were increased by IR. The focus of the studies are on hematopoietic stem cells, dendritic cells, and most importantly, on vaccination responses. Progress made thus far. The first year and a half was dedicated to the miniaturization of in vitro assays for both stem cells and DC quantification and differentiation potential, a program that is very nicely useful because it will be very useful to researchers looking both at pediatric and geriatric populations outside of the radiation research. There's been also an establishment of long-term, and I underscore long-term, mouse models for the longitudinal study of the effects of radiation in aging on the function of immune murine cells as well as human immune cells by the use of humanized mouse models. And I just already mentioned influenza vaccination studies, which is first ever in A-bomb survivors. And the publications are starting to come out now in the fourth year of operation, with 6 peer review publications already out, 3 submitted, and 17 in preparation, with more to come. So I'm going to talk very briefly about the predictive biodosimetry element of the portfolio. It is a program that was just established, barely a year into its existence. It includes 6 grants to Dana-Farber Cancer Institute, the Hutch, Georgetown University, Medical College of Wisconsin, UCLA, and University of Rochester. The objective is to identify and characterize biomarkers that predict radiation injury to specific organs. And beyond understanding at the very beginning what a radiation dose might be and use that to guide our treatment, we would definitely want to know what the long-term effects might be in the physiological system, and to get a better understanding beforehand of which organs might be affected by what type of radiation and the degree of radiation. And the biomarkers that are under analysis right now are all linked to relevant clinical outcomes, and they are strictly related to radiation exposure and, most importantly, stable at 24 hours. We cannot look at ephemeral markers. They need to be present at 24 hours and stable in order to be able to be tested in a field situation. And with this particular slide, I finish my presentation, and I'll be happy to try to field any questions you might have.
I think we can take 1 or 2 questions.
Okay, thank you very much.
Okay, mouse. Oh, that's me. I am David Cassett, and I will be talking to you about focused product development, and hopefully tell you what focused product development really means. And you've seen this slide before, and you'll see it again, but I just want to point out that there are red boxes here that show you what my universe is, and so I will go into more detail in the next slides. So what is focused product development? So in 2005, there was the strategic plan that was put out to direct NIAID in our product development and going towards countermeasures against radiation. And there are several elements of this. The first is to really look at gaps and find compounds that we could fund in order to accelerate their development to get to products that were needed for the strategic national stockpile. Among these were novel decorporation agents and oral DTPA. So the idea here was that if somebody gets radionuclide inhaled or in a wound, you want to get rid of the radionuclide. We have countermeasures against gastrointestinal ARS, and also GCSF, Neupogen, Neulasta. These are the filgrastim, pegfilgrastim, leukocyte growth factors, which were the low-hanging fruit, and we'll get to that later. The other part of that I have is the product development support services. So the companies that have products that look promising for radiation, we would be able to accelerate their development so they could get to the IND, investigational new drug stage, and then hopefully licensure. But what we had to do is to build up the capabilities, sort of like our own pharma company, develop and refine animal models and test the countermeasures. And by we, I mean I just push the papers and approve the invoices, but it's really to contract out for another organization to be able to do this. So the first set I'll talk about are the novel decorporation agents and oral DTPA. So here, you want to give drug to people who have radionuclide inside their systems, because radionuclide, in giving radiation inside your body can produce long-term effects. There is a product that is approved for this, DTPA, but it's given IV daily, which is not suitable for mass casualty situation. Instead, you want to give these products orally. So we awarded a number of grants and several contracts, and right now we have 3 products that have been reviewed by the FDA at pre-IND meetings, and that we hope to get INDs out of these. In the interest of time, I'm not going to go over the data for these, but they're very similar in that these products, three different ways of approaching it. One is a new drug. Second is a prodrug, which was metabolized to DTPA. And the third is DTPA with additives that will allow for increased absorption in the GI tract, because DTPA itself is poorly absorbed. And the bottom line for these is that they are as effective as DTPA given intravenously. However, you could give these orally. So these are much better for hundreds of people. And these are the data I'll flash through. And go to our product development support services contract. This was an RFP that was awarded in 2005 and 2010 to the University of Maryland School of Medicine. And they've set up a consortium of them plus several subcontractors that can perform a number of studies that are in product development. So you want to have IND, FDA IND, product development leading to submission package to the FDA. And these include the support services. So these, not just efficacy studies, but any kind of studies that could bring a drug down the pathway to an IND. So we look at various decorporation on ARS treatments. And this is a blow-up of the product development pipeline. The product development contract pretty much takes the middle part of this, although we go back to the animal model development because you need the foundation in order to accelerate products this way. And we've bled into the advanced stage development as well, but pretty much we're in the mid-range, so we try to get companies come to us, they give us that we can test their compounds and hopefully get them from here to here. So what we've done, under the contract, is part of it is the foundation. This is the infrastructure. These are facilities that are GLP, good laboratory practice, or generates lots of paperwork, compliant. Also, facilities for radionuclide studies, so that the contracts that I talked about earlier, they can go in, once they have an IND, they can be tested under GLP conditions. Also, to bring everything together, you need some core facilities. You need dosimetry, statistics, because you want to have your animal studies sufficiently powered. And also, there's a large number of data to manage and document management so that we can have crosstalk among all the different elements of the contractor. We've also managed the animal model development in hematopoietic, gastrointestinal, and lung models in mice and in non-human primates, and also the radionuclide exposure models. And this is the important point, that these models are relevant for research or approval under the FDA animal rule. And if you're not familiar with this, Bert has pointed out that this is a way for drugs to be approved without giving it to irradiating people and giving them the drug. So you can't lethally irradiate people. But you have to have very strict guidelines. One is that you have to have an endpoint that is relevant, which they define as mortality or major morbidity. Second, you have to have well-defined studies in 2 or more animal species. So here we have animal models here and GLP-compliant facilities. Third, you have to understand the mechanism of action of the injury, the radiation, and the mechanism of the drug. So we designed these with endpoints, secondary endpoints that speak to that. And third and fourth, you have to be able to get the dose right that is relevant for people. So we perform pharmacokinetic and pharmacodynamic studies. And the animal models that were developed were published in 2 special issues of Health Physics. And what this is nice is that when somebody comes to us with, we have a promising drug, what do? We can say, look up the model development papers in the October Health Physics. Or the FDA is also interested in the animal model development, so they can look, they have these articles as well. And with the FDA, we also have the interactions with the FDA that inform us of what the FDA is expecting. We also give information to the FDA so that they can use that to apply to anybody who comes to them. And our products, our accomplishments are as we have a number of medical countermeasures tested in the product development contract for the heme ARS, GARS, ARS, and also upcoming in the lung models. We've identified several of these candidates for further development in mice and non-human primates for efficacy and pharmacodynamics. We have 2 PIND. A PIND is like an IND, but there's no clinical protocol, submitted to the FDA to support late-stage development. So this farther than we intend to put people or put products, and 2 pivotal heme efficacy studies. So I'd like to finish up with this. This is the story of low-hanging fruit. So when I started the program in 2004, we were already having discussions with Amgen, the producers of Neupogen, filgrastim, GCSF, the countermeasures group at the FDA, University of Maryland, and NIAID. And the idea, even though Dr. McBride said that GCSF may not be the best thing, it is in the shelves all around the country. So that's an advantage. In our discussions with them, we wanted to develop this into a licensed drug. And we looked to see whether we could increase the survival in lethally irradiated monkeys. Now, the issue here is that the company pulled out before we could do the pivotal study. So we pared it down. Instead of 3 radiation exposures, we went to 1. But it was a GLP study. So the idea is to give GCSF or Neupogen every day starting at 1 day after radiation. The animals also receive antibiotics, fluids, nutritional support, blood products as needed, and to see whether the animals would survive. So the first question is, did they survive? Yes, because I wouldn't be here showing these if it was a failure. We increased the survival from about 40% up to about 80%. So we did see an increase, a significant increase in survival. What's important is that this survival was linked also to the recovery of neutrophils. So here's the control, here's the animals that got the drug. So we were very happy because now we have a real study, GLP study. We sent this off to the FDA and thought that if nothing else, we could inform people who would be using the drug in case of emergency, or the FDA could issue an emergency use authorization. So we had, high hopes. But what actually happened is that there was interest in the leukocyte growth factors from the FDA. So the FDA called us and said, could you present the data on your drug? And we will convene a joint meeting of 2 different advisory committees on the imaging and oncologic drugs to see whether these data could be used for a license. So the question that they put to the panel was, with this animal data and with the data from people, efficacy and safety data and pharmacodynamic data, is this reasonably likely to produce clinical benefits in people who get exposed to radiation? And drum roll, please. The answer was yes. Overwhelmingly yes, 17-1. So this is like more than we had ever imagined. We were supposed to have poker faces because we were in front of everybody, and we did, but we were kind of leaping inside. So this is, so the company can now take this drug, put their submit their data as well, and there may be a chance for an approval. But that's not the end of it, because daily dosing is not as good as giving just, say, 2 doses. So Amgen makes another drug, Neulasta, which is longer lasting, Neulasta, to increase survival. So we did another GLP study, 2 doses of drug, starting one day after radiation, medical management, and got a very similar result. So here we're going from about 50% up to over 90% survival. And again, this is linked to the advanced neutrophil recovery. So in conclusion, we've accelerated the development of specific medical countermeasures. We built up a foundation for our product development program, and we're taking in drugs. Even now we're in negotiation with companies and planning protocols for these and other organizations. So one of the new initiatives that is coming up this meeting is the third iteration of the Product Development Support Services contract. And I'm done. Any questions? If not, let's see.
I only have 3 minutes to get us back on schedule. I don't think I can quite manage that.
Where says Andrea?
Okay. I only have 6 slides, so it should be relatively fast. So I'm Andrea. I'm a program officer in RAD-NUC. This is my part of the RAD-NUC universe, as you've seen previously. The only thing I would say about this slide is that this represents a continuum. We have the basic research, and we like to apply what I consider a vacuum or a negative pressure to the researchers in our portfolio, pulling them forward into some of the more advanced product development. We do this kind of in a stepwise fashion. We have the early products that are being studied in the medical countermeasures arena in the U19s that Francesca spoke about. Then we try to pull them into these intermediate tissue injury and biodosimetry grants where we provide additional support and guidance, and again, urging them forward. A number of those grants have actually transitioned to BARDA, and we continue to serve on coordination teams on those BARDA contracts to provide a historical continuity and guidance to BARDA, because we were the agency that funded the early research in those areas. And then you can see here under contracts, we like to then go into a contract mechanism where we can have additional control over the product and be able to share our experience that we've had with the FDA, with the investigators, again with the idea of just accelerating them to this end goal, which is the licensure of their product. I'm also involved in a number of different agreements that we have with both NIH agencies and outside, and we have an exciting partnership that was just initiated with the Institut Recherche Sécurité Nucléaire in France, which I'll briefly touch on as well. So you've seen these slides previously. This is our standard quad chart, and I tried to pick drugs that were already available clinically for other indications to highlight for you, because we, again, feel these are the low-hanging fruit. This particular drug, interleukin-11, opralvacin, which is also known as Numega, some of you may be familiar with, it is used in chemotherapy-induced thrombocytopenia, but clinically it has some side effects when given systemically. They did, however, note in some of these early clinical studies that there was an improvement in GI damage in some of the both patients and preclinical models they were studying. So in studies done by Dr. Sandy Burnett at University of Arkansas, in a rat model, he's actually giving the drug orally. And what I find is exciting is that when I started applying this pressure to him, he actually went to Pfizer, who is the parent company that oversees this Wyeth market product, and he was able to get them to come on board with him and is now starting interactions with Pfizer. But as you can see here, we're up in the 9-grade dose range, which is a typical low GI, high hematopoietic dose range. And you can see that with this drug given orally by gavage in the rats, you can get a 50% improvement in survival by 30 days. Now, he's not carried these studies out later, as I know a question was asked previously. But we're very excited because we're used to dealing with small biotech companies, and Pfizer now, and then the recent interactions with Amgen are getting us into these companies that clearly have regulatory support. So there's less that we have to provide to these early investigators. And the next drug I'm going to highlight is enalapril, which is an ACE inhibitor, the angiotensin converting enzyme, which, as you know, is a commonly prescribed antihypertensive drug. This particular study was done in rats, and it was done by me Theodora at the Medical College of Wisconsin. The animals were given 13 gray, but only to the whole thorax, because a 13 gray TBI, or total body exposure, would not result in any sort of late survival. And the thing that I think is most important about these data is that not that there was a 40% improvement in survival, which is significant, and you're going out to 180 days, which is where you're really seeing this intense lung damage, although it begins at day 70 to 80. but that she didn't initiate this treatment in the drinking water of the animals until 35 days post-exposure. So that's very exciting. People who do these late effects have a huge advantage over the people that are studying the GI and heme, where you die within a week or two, in that the deaths tend to come several months out. So this is particularly exciting just because in the aftermath of a radiation event, it's going to be difficult to get a drug into someone quickly. So this 5-week start time post-exposure is really exciting. Just to highlight the grants that we co-fund with NIDDK, they are doing the Intestinal Stem Cell Consortium, which I believe Dr. Rothermel pointed out to me years ago, and we signed on to their RFA. And this is exciting because what these basic researchers are doing is trying to make GI stem cell transplants as commonly carried out as bone marrow transplants are now. And for obvious reasons, we signed on to this because of the known damage to the gastrointestinal tract following radiation exposure. They were awarded in 2009, and they're recompeting right now. They're being reviewed. And they are doing a number of different studies, but primarily what they're doing is taking the stem cells out, culturing them, and trying to put them back into a mouse model. And as a side aim, a number of the studies happen to use radiation in order to denude the GI tract or to initiate the damage that they then want to repair. So you can see here, this is unpublished data, so I've de-identified it. But you can see when the animals are not irradiated, you get a very nice stem cell crypt structure, nice villus height. It's a very healthy in block A, unirradiated. But when you irradiate the animals with 12 gray, you can see a tremendous difference in the histopathology. This is a jejunum section. And you get a loss of crypts, and you see this blunting of the villus and a thinning of the submucosal layer, which when this drug is given 24 hours after exposure, you get a very nice recovery of the GI tract. So this has been a very nice interaction that we've had with NIDDK. The other thing I'd like to point out is that we actually fund some intramural studies that are being done through the National Cancer Institute, because many of the researchers there are not only focused on cancer, but always focused on radiation damage. And we're doing this because they, for example, this particular study is a biodosimetry study, although we also have epidemiology work that we're funding, as well as some medical countermeasures for late effects. I chose this one, and you've heard about the gamma H2AX foci. It's a phosphorylation of a histone protein that happens at the site of double-strand DNA double-strand breaks. Sorry, trying to go too fast. And what they've done is they've taken lymphocytes from human blood and also from mini pigs, and they've ex vivo irradiated them and then looked to see the incidence of these foci picked up with a fluorescent antibody. And the take-home point here is that in comparing the human and the pig, they find that the responses, these DNA damage responses, are very similar, which means that this is a nice biomarker that could be used to link or to bridge an effect that you're seeing in a preclinical model, which is what you can test in for the animal rule, as well as a human response. So that's why I selected this particular one. And you can see that although the signal is fading, it is still present at 24 hours, and other studies have shown that it does continue beyond that point. And this is my last slide to go a little clinical, and I apologize if you're squeamish, but this particular study is important because we just signed a statement of intent with this group, the IRSN in France, and they are currently clinically working with patients who have been exposed to radiation following industrial accidents. They actually are contacted frequently, and in fact, the patient in New Mexico from that particular source accident they were called on to consult in those cases. So they're working with mesenchymal stem cells, and we happen to find out that the FDA Center for Biologics and Evaluation and Research is also interested in mesenchymal stem cells in characterizing them. So what we've been doing is trying to broker an interaction between our group, the IRSN in France, and also the CBER group, so that we can try to better identify the purity of these mesenchymal stem cell populations. You can see here clinically, this particular patient was injured in 2006. They were holding onto a radionuclide source, and they had some cutaneous, rather striking cutaneous burns from the radiation exposure. And with the presence of these injections of these mesenchymal stem cells, they saw a dramatic improvement. They were much healthier, and they've been following these patients out, and they've not seen any relapses, which tends to happen. Radiation is the gift that keeps giving. You have a apparent healing, and then it decays again, and then it heals, and then decays. So this is rather exciting, and this particular patient was in 2006, but 8 patients since then have now been treated with mesenchymal stem cells. So both Dr. Rotrosen and Dr. Jacques Rupersard have finally signed the paperwork, and we're off to the races on this very exciting new collaboration. So sorry I went quickly, but does anyone have any questions? Great, thank you.
In the NCI study, you highlight the fact that the pig cells look like the human cells, but so what about smaller animals?
Yeah, so in fact, and also in non-human primates and in mice, they have seen similar, like the lines are almost on top of one another. So this particular study is just consistent with those that are being seen for non-human primates and also for mouse. Well, pigs are becoming a more sought-after model in radiation exposure because the non-human primates are so difficult to obtain, and there's the concerns about using these very special animals, whereas there's less of a concern using, for example, a mini pig. They're easier to take care of, and they're just starting to understand the responses of the mini pig and see if it can be a useful model going forward, as opposed to a dog or a monkey. Yes, pig skin is excellent, and actually the FDA has been advising people that they would like to see pediatric studies in mini pigs. So a greater understanding of the mini pig structures and how they respond to radiation can help link that model and make it more user-friendly, more applicable to these advanced development studies.
Okay, good afternoon. My name is Carmen Rios. I'm a AAAS Science Policy Fellow and a Program Officer, and I've been with this amazing team of people for about a year and a half, and they've been keeping me busy. So what I'm doing here with this program is I'm working with the SBIR program, specifically Program Announcement 12044. I'm serving as the Program Officer on that. I'm also the company liaison, so I'm the point of contact for companies that are coming into our drug development program. So I'll set up meetings that are interagency meetings involving the FDA, BARDA, DOD. I'm also a product manager, so once we bring them into our program, like all the other people on our team, we're assigned drugs that we follow and guide through our process. I'm also the point of contact for the AFRI study. So David mentioned our product development program. So once companies come in and we find that they have a promising agent, we will perhaps put them through our drug efficacy screen. So it could either be through AFRI or through MCART. And we provide guidance and follow up on that. But today I'm going to talk to you specifically about the SBIR program. And basically, as you know, the Small Business Innovation Research Program in general is to encourage small businesses. And for us, we're interested in getting people to focus on radiation and nuclear development to prepare for IND submissions to the FDA. And the funding supports non-clinical, preclinical product development for future clinical safety studies, GLP animal model efficacy studies, and eventual FDA licensure. So just a little background, and David touched upon this. There are really very few licensed products in the Strategic National Stockpile. Mostly they're decorporation agents. So there's really a critical need to focus the research on radiation and nuclear drug development. And so our priorities include mitigators that can treat ARS and the delayed effects of ARS, decorporation agents, and biodosimetric tools that determine radiation exposure that serve to triage patients during a mass casualty radiation or nuclear incident. So SBIR application types include phase 1 and phase 2. Phase 1 studies will help to establish technical merit, feasibility, and potential commercialization of a particular testing agent. This will help to determine the quality of performance that will hopefully move into phase 2. Possible funding is $300,000 per year for up to 2 years for phase 1. And then for phase 2, where continued efforts can be made in research and development that were first initiated in phase 1, they can receive $1 million per year for up to 3 years. So there's the potential of $3.6 million that are available through our program. So before a very specific program announcement was created, for the SBIR program to meet our needs, there was just the NIH Omnibus and the NIAID Advanced Technology SBIRs. And you can see the numbers appear fairly low. For each, at least 1,200 applications were received during this time frame, and a total of 2 RAD-NUC applications were awarded from the Omnibus and 5 for the Advanced Technology SBIR. So once the awareness became or grew for the radiation and nuclear need, the specific program announcement PA 09093 was established, and out of 104 total applications, 11 Phase 1 awards were given and 3 Phase 2 awards. And then the renewal of that program announcement under the number PA 12044, there were 3 Phase 1s awarded, and there are actually 23 still under review, so we'll see what the totals will end up being on those. So just to highlight some of our grantees, I have 2 companies that I'll highlight. This is Boulder Biotech out of Colorado. Andy mentioned IL-11, hematopoietic growth factor, but this one's a little bit different. It's a pegylated form, so it's, a longer lasting. This is a mouse study that they were going to do PK/PD analysis for sub-Q and IV administration. And the aim of this program, or this application, was to demonstrate the feasibility of using this novel long-acting pegylated form of IL-11 to accelerate platelet recovery and to improve survival in ARS mouse model. And they were able to show that just from a recent publication out of the Health Physics 2014. With only 1 administration of pegylated IL-11 at 1 milligrams per kilogram, they saw 83% survival, and at 0.3 milligrams per kilogram, 90% survival. So ordinarily, IL-11 in the non-pegylated form requires repeated dosages, and so it's just not feasible after a mass casualty incident. So this is something that's much more appealing. And that was also shown with the platelet recovery starting at 10 days that supports the usefulness of this agent. And just their status now is that they just recently entered or were awarded the NIAID U01 for medical countermeasure development, and so the work continues with this agent. A second company, Terapio, who has been quite successful in our program, is looking at RLIP76 protein liposomal formulation. And this is interesting because this is an agent that helps to counteract the sequelae of free radical damage that Dr. McBride mentioned earlier. And it's a transmembrane protein that actively transports products of lipid peroxidation out of cells. And so they were able to get through Phase 1 and Phase 2 studies through our program, and so they were able to establish protein stability in Phase 1 and the route of administration and dosing. And then in their Phase 2 studies, they optimized their proteoliposomal formulation, which makes it longer lasting, and they developed assays for IND-enabling studies. They were able to show 41.2% survival after 6 doses of RLIP76 in mice that received lethal levels of radiation. And this company is interesting because they're really taking advantage of all kinds of funding mechanisms that we offer. One of the things is that they are in a program called Bridges that you may be familiar with, which is sponsored through NCATS, and we are co-funding this program with NCATS. That's the Bridging Interventional Development Gaps program, and this will help in the scale-up of RLIP76 for future IND submission. And the final slide is just to show you an example of a drug development profile. This one happens to be from Terapio, but it's a commonly seen program, but just to show you how they've really patched together all of the opportunities that we provide. So if you can just follow the purple squares there, starting with model characterization and efficacy in mouse and NHP models. That was through our drug development program that David mentioned. They also have been receiving support through our SBIR program, which I mentioned, starting in 2009. So through the SBIR program, they've been able to do IND-enabling tox and PK studies. And in yellow, you could see their own company support, which has been interesting because we have a continual dialogue with the company where we're able to look at comparisons between our data to help provide guidance. And then now they're entering the Bridges program for final scale-up of the drug, so they can continue to move on into an IND submission. And that is all that I have. Any questions? Okay, thank you.
Kind of a general question. There seem to be some nice, impressive results with a diverse set of products that are showing efficacy, and it looks very nice. Are there any efforts or capabilities of understanding the mechanisms of action of these products in conjunction with the CMCRs? Is there back and forth with the more basic scientists? I think many of these products look like they have an obvious mechanism, but you can't assume, and some of them you may not know. So is there a connection there?
Okay. Mechanism of action is extremely important because FDA does not have human efficacy data. So if you have a mechanism of action that you can show with strong data in more than one species, that would indicate that that mechanism would be conserved from species to species and then hopefully to humans. If you can make that link, it makes it a stronger case that it would also potentially have efficacy in humans. So mechanism of action is extremely important. The other aspect of that is you only see what you look for. As you were saying, many drugs have more than just one action. I mean, like tetracycline we're finding out, and doxycycline, and other things that Bill has been testing may have more than certainly one indication. One of the things you'll hear in my next presentation is the exact point that you're making, the stronger collaboration of other elements of our program with the CMCRs. And using mechanism of action as one of those things to help that is certainly one.
Yeah, I'd just like to say, in addition, that some of these compounds did go through some of the CMCR centers to begin with, and then they kind of disappear, and sometimes you don't hear of them for a while, and then they come back again. But I do think there is probably a need for doing more of it, but we do see some of these compounds and do some of the very preliminary work that leads to their further development. And you kind of pass them on and hear about them later.
Okay, before we start, we've been here for a long time sitting. Why don't we all just stand up? Get a little shake and get that blood moving and, well. Okay. Wait a minute. No, once I get on a roll, you know that. Okay, well, I'm here to tell you about three concept clearances that we'd like to obtain council approval for today. Two of them are renewals, and one of them is a brand new one. The renewals of our Centers for Medical Countermeasures Against Radiation is the first one I'm presenting today. You've heard an awful lot about the CMCRs, as we call them, from Bill and from Francesca. But this is a renewal of our U19 cooperative agreements, and we anticipate this next round that we would have about 5 to 7 awards, and these awards are for 5 years. The background is, you've heard, so we want to support basic research, translational research, and development of medical countermeasures, and also biodosimetry devices for diagnosis of radiation injuries, and in support of medical management, and then also be able to have drugs to treat these types of injuries. And we want to continue to expand that program, or continue that program. I wish I could say expand. Budget situations being what they are. As you know, the program was established in '05. We had 8 centers at that point. We recompeted in 2010, and we have 7 centers currently, and the new recompete of this program, we hope to have 5 to 7 centers. I think you've seen this slide before. This is just the geographic distribution of our current 7 centers. Accomplishments of the CMCRs, I think we also had quite a bit of information on that. If there's anything different here, no, I think Francesca covered this fairly well. Lots of publications. To me, publications mean creativity. And then also there's patents that have been provisionally submitted for, and that shows productivity. So creativity and productivity are a hallmark of these centers. And then also, as Bill said, he has 100-plus people in his center. Among the whole program, I think we probably have, I think my count at one point was close to 2,000 people that our program in total affects. And so there's a lot of smart people, thank goodness. The requirements for this new program are going to be that it's a 5-year award. The requirements are also that the CMCR has to have proposed 2 major synergistic projects in basic translational or applied research for a medical countermeasure. They also need to have core facilities, and the core facilities would be dependent upon what they need to support their research. And then also, what I really like about these programs as well is they have a pilot program to generate new ideas and support funds for preliminary research to gather data so that some really wild ideas can come out and hopefully be successful, and then be successful in using that data for an R01 or other type award. And that's a 2-year program, possibly with a maximum of $75K per year. So our future programmatic activities that we want to prioritize here, as we've maybe alluded to, is that we want to harmonize and maximize our resources to minimize our costs within the CMCR program. I think Bill alluded to one of the PIs from the Duke CMCR program is now going to UCLA. He's doing a very good job at recruiting. But actually what that means to us is that there's a lot of interaction among the different folks within the CMCRs, and we want to continue to encourage that. We want to integrate and engender collaboration more so with the product development aspects of our program, so that we have more of a seamless program across the board, and that we can take advantage of all of the extant expertise. And then obviously the emphasis is going to be on advancing promising candidates towards IND, so supporting more IND-enabling kinds of studies. We've been in this program for close to 9 years at this point. When this is recompeted and awarded, it'll be 10 years. It's about time we kind of make a little bit of a shift from basic research to more product orientation, and so this will be an effort to try to do that. Not saying we don't want to do research, because we always want to continue to do research in order to keep that pipeline going. So that's the CMCR program, and I'd like to get approval for that. So shall we just go on to the next, or the questions?
I guess I have one question. There was a brief discussion about understanding mechanisms of action, and as part of this, is there going to be something you plan to do to incentivize research in that direction that you think would be helpful?
Great question. I would probably want to understand what drug we're working with first. Each drug has its own unique set of mechanisms of action. And so to undertake just a general mechanism of action program at this point, I think I don't know enough to do that, so I would focus on a particular drug. If that makes sense to you.
Perhaps Arlene is asking a little more general question. Is there going to be flexibility built into the CMCRs beyond the pilot projects? Or would you use the pilot projects with a higher priority for those kinds of projects in order to address these issues as they arise?
Okay, the pilot projects are more driven by the investigator, but that's an interesting concept. I think if what you're saying is as we do with some of our other programs, we have purposed programs, and it's possible that we might be able to do some of that. Bill?
Yeah, I mean, I actually think that, as a PI, the UCLA CMCR, I know that we use our pilot program exactly like that. Often it really is taking some outstanding scientists who have, real insight into mechanisms and who have the ability to kind of take our products, and actually look at that. So it's a very important part of the pilot program for UCLA anyway. I can't talk to all the centers, but I think it's probably true, generally as well.
You could consider, as we do for some other programs, an opportunity fund, which is managed in common across the centers and is, can be defined, its purpose can be defined by a steering committee.
Excellent. Actually, we are thinking about incorporating that type of concept within the new competition here, Opportunity Fund, and also reorganizing a little bit of the structure of the centers such that we have advisory board that is common to everyone. That might be helpful there, too. Okay. Next concept up for discussion is the, excuse me. Oh, you want to vote now? Okay.
Council members, so how do we do this? We ask for someone to nominate this for approval. Ask for a second. Ask for all the yeas and nays. Anybody vote nay? Okay, it's approved.
Okay, the next one up for grabs here is the Radiation and Nuclear Countermeasure Product Development Support Services Program, or contract, that David Cassatt told you a lot about. We're looking for a renewal. It's for one award. The one award meaning one prime contractor. I don't think there's any one contractor who can do everything that we're asking them to do here. So the prime contractor would have a coven of subcontractors to cover the array of capabilities that they're going to need to have to support product development, which you'll see. Our current contractor has, 8 to 12 subcontractors, depending upon what time of the year, supporting all the different aspects of the different I&D enabling kinds of studies that we need to have done. We'd also have this one as a 5-year contract. This is to provide funds for more advanced product development, to support I&D development, so that we can get some drugs licensed to include into the Strategic National Stockpile. The current contract that we have is with the University of Maryland School of Medicine, and they have now had that contract for 2 iterations of this program. They call themselves MCART, Medical Countermeasures Against Radiation Threats, and that's their name for it. We call it the Product Development Contract. Among the things that they need to provide support services for are all of the things that are listed here and others. David mentioned a lot of administrative aspects, from dosimetry to data management, document management, GLP, auditing capabilities. So that's also part of this, but these are just the scientific and developmental aspects of what we want to have within the support services contract. We want to be able to evaluate candidates for efficacy. We want to be able to develop optimized routes of administration and dose forms for each candidate. So it's not only just efficacy, but it's also optimization of the drugs. We want to be able to study the effect of these drugs in the acute radiation syndromes, plural, as well as the delayed effects of acute radiation exposure, so the later effects, the fibrosis and pneumonitis type things. We have the group who wins this would also need to have extant models, rodent models for hematological as well as gastrointestinal ARS. They would need to have non-human primate models of hematopoietic ARS and develop models of hematologic problems such as thrombocytopenia, and in this case using a canine. One of the interesting problems we have in some drugs, some protein drugs, some peptide drugs, is that they're exquisitely species specific. There's one particular drug that is so specific to humans it doesn't work in any other species other than the chimpanzee, which we're not allowed to explore. So it makes it very difficult to try to find models of animal models that then would recapitulate the human pathophysiology and also drug response. So that's why we wanted to have the canine model as part of this as well. And then also continue the development of the gastrointestinal models in the non-human primate, since we now have some drugs and some concepts of products that are looking as if they actually will work in that high radiation exposure level for gastrointestinal ARS. More common, I guess, toward drug development would be support for good manufacturing practice, GMP material, manufacturing support, as well as stability studies. Got to do a lot of paperwork type toxicology studies, GLP toxicology studies, and farm safety studies, GLP-related pivotal animal efficacy studies as per the animal rule to provide at least the 2 species capabilities there for non-human primate, and in this case, rodents for ARS. And then also we're anticipating, because we're farther down the road of product development at this point, that we may actually need to initiate a phase 1 human clinical safety study or pharmacokinetic study. Even though we can't study efficacy in humans, the FDA still says, according to the animal rule, that you have to study safety in humans, and that's obvious, I think. So we want to be able to support activities in that area as well. We have a great group that we work with here within DATE, the Office of Regulatory Affairs, and they support us strongly for FDA submissions, for pre-IND and PINDs, and we want to continue to be able to work with them and provide them on our team as well. So our objectives for the planned initiative coming up, we want to have and establish more collaborations with the CMCRs. We want to refine and continue to develop these animal models that we spoke of. We want to seek FDA animal model qualification. This is a new program that FDA is initiating now. They're having a animal model qualification process, very akin to an IND type process. It's very organized, very prescribed, and we're just about ready to initiate our first letter of intent to engage them in this process. We want to continue to screen, evaluate, and optimize candidates, I think a lot more than we have in the past. Perform the kinds of IND enabling studies we need to have for IND and NDA or BLA support, and continue to support the pivotal studies that we need for the ARS and the hematopoietic and the GI ARS indications. And then I think we also want to have the ability to help these folks develop formulations, analytical methods to support formulations development stability studies that are useful in a mass casualty situation, and also useful in a mass casualty situation for all of the at-risk populations, from a newborn to elderly The kinds and types of routes of administrations and formulations may be different. A newborn, a 1-year-old, a 2-year-old may not be able to swallow a pill, so we want to be able to have formulations that are compatible with them as well. So we want to have more support for those kinds of activities. Again, support phase 1, clinical safety studies and pharmacokinetic studies, and then support the IND packages. And those would be the objectives for the new effort. You want to repeat this one? Oh. Okay. It's always good to have more eyes. Okay, this is the last initiative that we're going to be presenting today, and this is a new one. This is a new initiative for a contract, for a broad agency announcement type contract initiative for the development of medical countermeasures to treat and mitigate radiation-induced lung injuries. As you've heard numbers of people speak today about the later and delayed effects of radiation injury, certainly affecting the lungs primarily and affecting pneumonitis and fibrosis. There were a set of folks in Japan, in Tokumura, who were exposed to upper body radiation levels. Those folks were able to survive 90, 80 days. They were able to get through the hematopoietic issues that they had, but they succumbed to lung fibrosis and lung injuries. And so that's why this is, I think, a very important aspect of what we do. We now have some basic research that we've done that have shown some promising agents, as Andy has described and Bill has described. So we'd now like to take this a step further and provide more product development support activities. We would expect to be able to fund about 2 to 4 contracts here. The contracts would be for 3 years. It's quite interactive because we want to make sure these folks are focused on the path to FDA licensure. As I said, there are no medical countermeasures for this or any other ARS effect, actually. So this is important to be able to provide the pathway toward licensure, and that radiation-induced pneumonitis and fibrosis contribute substantially to late mortality and morbidity. So we do have a critical need to be able to develop, license, and stockpile these kinds of medical countermeasures. Past activities, which we've alluded to, and Andy has told you a lot about. So we have possible candidates here that look promising. And so we do believe that this program that we're putting forth here would be a successful program. We would anticipate about 10 to 12 applications. The objectives, as we indicate, would be to fund IND-enabling studies to carry more advanced product development toward licensure for these researched compounds. And the obvious administration requirements of post-24 hours at least, post-24 hours post-irradiation. And then also to be able to provide some funds and ways to elucidate mechanisms of action of injury and mechanisms of mitigation of the actual drug. So we chose the BAA mechanism here because there will be, as we said, 8 to 10 potential applications. Each one would have a different drug. Each one would have a different drug at a different part of the product development continuum, so we couldn't set up a set of statements of work because it would be each company would be different, or each drug offerer would be different. So the BAA mechanism provides us the best flexibility. We tell them what we want, what our objectives are. They would then write their own statements of work depending upon where they are in the product development pathway, as well as the particular requirements for their specific drug. So they write their own statement of work and provides the best opportunity for them to compete for these kinds of funds.
So we'd like to also ask your approval for this program as well.
Any questions? I have one question. So
Sure.
wouldn't this work be covered under the product development contract? Or why are you breaking it out with the focus on the lung?
Okay, in looking at the overall program, the product, we have CMCRs, basic research, product development. We have tissue-specific injury grants. We have the product development support services to carry us for further product development. None of those funds go to the company directly. So the companies get to a point and they're like, hey, they want their own money. They want more control over what goes on. They want their own pot of money to continue their development. This gives them the opportunity for at least 3 years in this program to get funds for themselves with
guess
our
the-
interference, if you will.
I guess the question is, couldn't you develop the product development program so that you could allow companies to further develop their products and then feed them into a pipeline? So you have some base, I don't know what I'm saying. You have some base services that are through like a parent contract, but then they subcontract out to companies to further develop their products to feed into that pipeline.
I hear your question, and it's a difficult administrative problem to do it that way. This allows us to directly fund the company, which also means we have direct interaction with the company to help guide and advise them as to where to go. Through the Product Development Support Services contract, it's a little more circuitous. It's a little more difficult to do exactly what I'd like to do, is to help direct these companies directly.
Can I just add that the lung models also are much less developed than things like hematopoietic ARS. And so I think the diversity of models is such that perhaps at this stage you don't want it to be too narrow. And I think I would have some concerns about that.
Good comment. Agree.
Just one comment. It gets back to this issue of molecular targeting that I mentioned earlier. So I was surprised, or maybe that's the wrong way to put it. I would encourage you to, in the product development scheme that you've put forward, to think more about drug design and molecular targeting as a way of going about this. Because, it depends on what your goals are. If you just want to repurpose and give biologics, that's one issue. But if you do want to get to mechanism and develop proprietary compounds and design new strategies that might be more effective for tetracycline or whatever, it'll be hard to do that without some sort of structure-based approach, I would think.
Yeah, one of the big additions to our CMCR has been a pharmaceutical chemist who's really world-class. And, we needed that, because we had to actually synthesize all of our chemicals from the libraries, because the libraries don't provide them, but also to make analogs and to look exactly at the point that you're making. Within that kind of array of chemicals I showed you, we can actually interpret a lot about what is the active moieties, basically because we have all of that information on an industrial database, and we can look at chemical structure and functions. We can do SARS, and we can do all of these kind of manipulations, and we have the ability to synthesize all of these analogs. And I think that's a very important aspect of the drug development process, and I know at least one other CMCR has got similar kind of capabilities. A very important point, yeah.
Okay, so thank you very much. So we'd like council input on this, on these two last initiatives. And just to remind you, we don't need necessarily a yes or no. If you have questions or other concerns or issues you'd like to further explore, we can defer discussion and approval for a later time. But so at the moment, why don't we ask the 4 council members where they're...
I would move to approve both concepts.
All approved. Thank you very much. This concludes the open session of the council.
Thank you very much.
Thank you very much.